Hi friends, welcome to my YouTube channel. The aim of today's video is to give you a lot of information on mitosporic fungi, their status and their teleomorphic relationship. These mitosporic fungi are a large and heterogeneous group of fungi whose common characteristics is the absence of a sexual state. Many of the pathogenic fungi, either in human or the plants, they belongs to this group. The fungi imperfecti or imperfect fungi, they do not fit into the commonly established taxonomic classification of fungi that are based on biological species concept or morphological characteristics of sexual structures because their sexual form of reproduction has never been observed. They are known as imperfect fungi because only their sexual and vegetative phase are known. Mitosporic fungi are also known as anamorphic fungi or residual fungi. Formerly they were known as deuteromycetes fungi or fungi imperfecti. They have some specific features, like these are septate mycelial fungi. They reproduce exclusively by mitospores or Conidia. They have asexual form of reproductions, means they produce their spore asexually in the process of sporogenesis. They are unconnected residual fungi, either lost their sexuality or adopted alternative to sex like parasexuality. They are not a monophyletic fungi, means of or relating to a single stock, specifically developed from a single common ancestors. Kingdom fungi, which is one of the oldest and the largest group of living organism, which is a monophyletic group, means all the modern fungi can be traced back to a single ancestral organism. This ancestral organism diverged from a common ancestor with animal about 800 to 900 million years ago. And the meaning of monophyletic means a single clade. That means mitosporic fungi are not from a single clade. That's why these are known as non-monophyletic fungi. Their DNA sequencing make residual organism within the group of teleomorphic fungi. Comparative analysis of the DNA sequences, it has placed mitosporic fungi as asexual fungi in the existing classes. There are a number of problems in taxonomic classification of mitosporic fungi. Although fungi imperfecti or the deuteromycota is no longer formally accepted as an taxon, many of the fungi it included have yet to find a place in the modern fungal classification. This is because most fungi are classified based on characteristics of fruiting bodies and the spores produced during sexual reproduction, and members of deuteromycota or the fungi imperfecti have been observed to reproduce only asexually or produce no spores. Mycologists who study fungi extensively, they uses a dual system of nomenclature which was permitted by Article 59 of the International Code of Botanical Nomenclature that governs the naming of plants and fungi. However, this was abolished in the year 2011, an update of the code. Under the former system, a name of an asexually reproducing fungus was considered a form taxon. For example, the ubiquitous and industrially very important mold like Aspergillus niger, which has no known sexual cycle. Thus, Aspergillus niger is considered a form taxon. In contrast, isolates of its close relative like Aspergillus nidulans revealed it to be in the anamorphic stage of the teleomorph. that is the scocarp or the fruiting body of the sexually reproductive stage of the fungus, which was already named Americella nidulans. When such teleomorphic stage is known, that name will take priority over the name of an anamorph that lacks a sexual reproductive stage. Hence, formerly classified Aspergillus species is now properly called Americella nidulans. Phylogenetic classification of asexually reproducing fungi now commonly uses molecular systematics. Phylogenetic tree constructed from the comparative analysis of the DNA sequences such as RNA or multigene phylogenesis may be used to infer relationship between asexually reproducing fungi and their sexually reproducing counterpart. With this method, many asexually reproducing fungi have now been placed in the tree of life. However, because of phylogenetic method requires sufficient quantity of biological materials, that is, I mean to say spores of the fresh specimen that are from pure, that is uncontaminated fungal culture, for many asexual species, their exact relationship with other fungal species has yet to be determined. Under the current system of fungal nomenclature, teleomorph names cannot be applied to fungi that lack sexual structures. Classifying and naming asexually reproducing fungi is the subject of ongoing debate in the mycological community. There are a number of organisms which included under Mitosporic fungi, and some of the genus has been shown with their structure in this slide, like Ascomycota, Rust, Alternaria, then Cladosporium, and many more. Italian botanist and mycologist Pier Andrea Saccardo, he classified fungi imperfecti into the order like Monilials, Melanchonials, Sphaeropsidales, and Mycelia sterila. The organism whose conidiophore and conidia are directly on the hyphae, these were grouped under Monilials. On the other hand, if conidia form in saucer or the disc-shaped structures, which are called acervuli, or in singular from acervulus, these were grouped under Melanchonials. On the other hand, conidia, if they are formed in flux-shaped receptacles, which is called pycnidia, and in singular form pycnidium, then these were grouped under Sphaeropsidales. And in case of Mycelia sterila, here the organism produced on the sterile hyphae or sterile mycelia. They lack conidia. The order Monilials under fungi imperfecti was again divided into different families like Moniliaceae, Dematiaceae, Stilvillaceae, and Tuberculariaceae. Likewise, Melanchonials was grouped into the family Melancholiaceae. Similarly, the order Sphaeropsidales of fungi imperfecti were further grouped into the different family Sphaeropsidaceae, Zithiaceae, Leptostomatiaceae, and Accipuliaceae. Saccardo in Xylus fungorum, which was the first system of classifying hypomycetes, which was prepared based on the morphology of the spores, he grouped this fungi imperfecti under 7 sections based on the septation and shape of mature conidia. Number 1 is Amerospori. Here spores are single-celled or one-celled, non-septate, spherical, ovoid to elongated. Followed by Didymospori. Here spores are two-celled, ovoid to oblong. Phragmospori. Here spores are two or many celled with septa by transverse septa. Or many cases these septa are pseudosepta. Dictyospori, these were muriform, that is transverse and longitudinal septa they will have. Then Scolospori, this type of spores are slender, filiform, then several celled, hyaline or colored. Followed by Helicospori, here spores are helical, curved, allantoid, hyaline or colored. Followed by Staurospores, here conidia are star-shaped and radiate or trifarate, hyaline or colored. In 1973, Geoffrey Claude Ainsworth, popularly known as G.C. Ainsworth, he classified fungi imperfectly into three classes. If the organism produces spores in Pycnia or Acervuli, they were grouped under the class Coelomycetes. But if the organism produces sterile hyphae, that is Mycelia sterilia, and the genera that bears the conidia on the hyphae, they were grouped under another class that is called Hypomycetes. And all the imperfect yeast were grouped under Blastomycetes. During 1910 and 1911, Jean-Paul Volumin, who was a French mycologist, he studied various modes of origin of the conidia. He stressed conidia be the distinguished by their origin. I mean to say, he stressed more and more on the conidial ontogeny for classifying a particular organism. In 1953, S.J. Hughes, who actually proposed a scheme of classifying of the Hypomycetes based on the conidial ontogeny that is called their origin. He divided the fungi into 8 sections representing particular mode of conidial development. He was the man who has given the system of classification based on conidial origin, otherwise it is also known as Huggesian's classification. And according to S.J. Hughes, there are different origins of the spores type. Number 1 is blastospores. Here conidia born as a buds or blown out tip. The genus Torula cladosporium, they produces blastospores. Followed by gangliospores. Here conidia developed from the swollen tip of conidiophore. There are a number of the genus that produces gangliospores, and the two examples is Scopulariopsis and Amblyiosporium. Followed by porospores. Here single or whole of thick walled conidia formed through one or more small pores in sporophore wall. The genus like Alternaria, Carvularia, Helminthosporium, they always produces porospores. Followed by phylospores. Here conidia born on short unicellular oval or subcylindrical or flux-shaped phyllids with distinct base cell swelling or narrow distal. The genus like Aspergillus penicillium, they produces phylospores. Another is arthrospores. Here conidia are formed by breaking of hyphae into cells. The genus like Geotrichium always produces arthrospores. According to Dorfeld, during 1989, he classified Hypomycetes into the order Agonomycetales and the family Agonomycetaceae. On the other hand, the order Moniliales was grouped into 4 families: Moniliaceae, Dematiaceae, Stilbilaceae and Tuberculaceae. Similarly, the class Coelomycetes was grouped into the order Melanconiales and the family Melanconiaceae, and the another order Speropsidiales, which was grouped into another family, that is Speropsidaceae. Presently, the Kendrick and Carmichael in 1973, then Hawksworth 1995, Quirk and co-worker 2008, they divided the fungi imperfecti as imperfectist, formerly which were known as blastomycetes, then hypomycetes, anamorphic fungi, formerly which were known as hypomycetes, then acervular imperfect fungi, formerly known as melanconials, and last one is pycnidial imperfect fungi, formerly known as Sparopsidals. Based on the conidial shape and septation, Saccardo grouped this organism into A, B, C, D, E, F, Z, and it was further have two groups, that is 1 and 2. If we see in the group A, that is one cell, that means all were known as Mitosporic. Here Conidia may be hyaline or the bright, that is halo, or Conidia may be pigmented or dark, or the phaew. So if single celled spores of the Conidia were hyaline, then these were known as hyalospore, and if colored, phaeospore. Similarly, for two-celled spores, that is didymospore, hyalodidymy and phaeodidymy. Say 2 to many celled or multiseptate Conidia, these were phragmospore, or if they were hyaline, then hyalophragmy, and for pigmented one, these were phaephragmy. And for group D, that is muriform, that is dictyospore, and if they were hyaline, then hyalodictyi, or if they were pigmented, then these were grouped under phaeodictyi. Similarly, for filiform, scolospore, helical, helicospore, branched, stereospore, and so on. Then, on the basis of conidial or conidiomatal type and conidial spores groups were made. These were either hyphal or cinematal or sporodochial, pycnidial, pycniothydal, acervular, capulate, then stromic, as well as sclerotia. Through this chart, I am going to show the classification of the fungi imperfecti at genera level. So this fungi imperfecti is classified as imperfect yeast, and under these three genres are included: Candida, Cryptococcus, Sporobolomyces. The second class, Hypomycetes anamorphic fungi, here number of genera are included, starting from Alternaria, Botrytis, Sarcospora, Carvularia, Fusarium, Helmithosporium, Lemonaria, Monilia, Nagrospora, followed by Pyricularia, Rhizoctonia, Trichothecium, and Verticillium. The third group, that is acervular imperfect fungi, the genus like Coletotrichum, Pestaloiopsis, Spaciloma are included. In the last group, that is pycnidial imperfect fungi, the genus like Ascochyta, Lesidioplodia, Diplodia, Phylosticta, Phoma, Phomopsis, Septoria are included. I am going to show you a chart of anamorphic genera and their teleomorphic, that is perfect stage. So let us start with the yeast genera. The Candida albicans, it is the anamorphic stage, and its teleomorphic stage is Saccharomycetals. Then Cryptococcus neoformis, its teleomorphic stage is Phylobasidiala neoformans. Then Sporobolomyces salmonicolor, it is the imperfect stage, and its perfect stage is Azospora salmonicolor. Next genera is acervular. Here Glomerella is the perfect stage of Coletotrichum. Then Pestalosperia is the perfect stage of Pestaloiopsis, and Alcinoi is the perfect stage of Spaciloma. Then the pycnidial genera, here Didymela is the perfect stage of Ascochyta, then Botryosphaeria is the perfect stage of Diplodia. Similarly, Didymela, Diaphorte, Guigneridia and Microspirilla are the perfect stage of Forma, Formopsis, Phyllosticta and Septoria respectively. Under the Hypomycetes genera, here Livia, Comoclathris and Cosliobulus. These are the perfect stage of Alternaria. Then Cosliobulus sativus is the perfect stage of Bipolaris Sorokina. Then Botrynia is the perfect stage of Botrytis. Then Microspirilla and Spirulina are the perfect stage of the genus Sarcospora. Then Cosliobulus is the perfect stage of Carvularia, followed by Paranophora is the perfect stage of Drusillera. Gibberella, Nectaria and Calonectaria are the perfect stage of one genera, that is Fusarium. Then Molinia, then Cuscia, Magnaphorte and Thanadophorus. These are the perfect stage of Monilia, Nigrospora, Pyracularia and Rhizoctonia respectively. So my friends, thank you for watching my video. I hope you like it. And to get more and more knowledge on fungi, please keep watching my YouTube channel. Thank you.